Hi there. In this video, we're going to see a bypass, a non-canonical TCA cycle that we don't do, but many microorganisms, including some very important pathogens, can do. And we'll see, it's kind of interesting from a biochemical perspective. You see why they do it. And so let's get to it. It's called the glyoxylate cycle. It's a bypass. So normally, this is a TCA cycle, going clockwise as we have described it. But anything, all these reactions in gray, so 1, 2, 3, 4, 5 reactions, don't happen in the glyoxylate cycle. So we don't generate this 5-carbon unit and all these reactions here. So we start from a 6-carbon unit, isocitrate, which gets split into 2 compounds, a 4-carbon compound, succinate, and a 2-carbon compound, glyoxylate. Glyoxylate then joins, which is a 2-carbon unit, as I said, joins with another 2-carbon unit, acetyl-CoA, okay, which is generated by pyruvate dehydrogenase, and generates a 4-carbon unit, malate, which can then go on and do the things we have discussed in the canonical reactions of the TCA cycle. So why do they do this? Well, because they can make a 4-carbon unit, such as oxaloacetate, which is extremely important for biosynthesis, okay, for many amino acids, for example, from 2-carbon units, from as long as they can keep feeding the whole cycle acetyl-CoA, which is the classic 2-carbon unit that enters in the cycle, and then they can make 4-carbon units. So it's really a biosynthetic capacity that these organisms have that we don't, that they can actually generate higher order carbon units from 2-carbon units here. And so this is special. The key enzyme is isocitrate lyase, the enzyme that splits isocitrate to succinate and glyoxylate. It's a very simple reaction. Okay, it breaks a carbon-carbon bond, and you will see this right here. Okay, this is how it's happening. This is isocitrate. Remember, citrate had the hydroxyl group. Excuse me, the hydroxyl group on this carbon, but isocitrate. Excuse me. The hydroxycarbon is moved one position over, and this is where so it's this hydroxycarbon that will make a carbonyl, and so that this compound will break. This bond will break between carbon-carbon, and so we're going to get glyoxylate, which is this part shown here, and you know, the 4-carbon unit. So after that, glyoxylate will join with acetyl-CoA in the reaction catalyzed by malate synthase to give you malate, which is a canonical now 4-carbon unit that will give rise to oxaloacetate in the next reaction shown here. The succinate can also be used for other things. That's one of the products, of course, of isocitrate lyase. So again, the reason for doing this is so that this organism can make oxaloacetate, and from oxaloacetate you can actually make glucose. You can go back up to gluconeogenesis, as we will see. And what this means, this 2 carbons to 4 carbons, is you can get fats, which when they are broken down, generate acetyl-CoA, as we will also see later in the class. And from fats, you can make oxaloacetate and then make glucose. So we do not have this ability, definitely not to this extent that's sent out by this reaction. So this is a big deal, right? I mean, you cannot, we cannot do this, but this organism can do it. And that concludes the glyoxylate cycle.